Picture this. You just finished a meal that landed about 1,000 calories past what your body actually needed today. Maybe it was a second plate you hadn't planned on, a dessert that talked you into it instead of the other way around, or just a portion that quietly got out of hand. Somewhere in the next few hours, a chain of events kicks off inside you that touches your liver, your muscles, your bloodstream, and a few organs most people never think about at all. The common assumption is that those extra calories just become fat, simple as that. The actual sequence is stranger, more layered, and honestly, more interesting than that one-line answer. And by the end of this video, you'll know exactly where that surplus goes, hour by hour, including a detail near the end that surprises almost everyone who hears it for the first time. The first thing your body registers about that meal isn't sugar, isn't fat, and isn't even calories. It's volume. As food fills the stomach wall physically stretches, and that stretching is picked up by a dense mesh of nerve endings called intraganglionic laminar endings, wrapped around branches of the vagus nerve that run through the stomach's muscle layer. When these endings sense tension and stretch, they fire signals straight into the nucleus of the solitary tract in the brainstem, a relay station that starts processing how full is this thing before a single calorie has even been absorbed. What's genuinely interesting here is that this early system doesn't care what you ate. Researchers established this decades ago using an elegant, if slightly odd, experiment on rats. They infused nothing but sterile saline solution directly into the stomach and measured how much food the animals ate afterward. It's worth being clear that this is animal research, not a human trial, but the pattern was striking. Meal size dropped in almost exact proportion to how much fluid had been added, even though the saline carried 0 calories and no nutritional information whatsoever. The signal was purely mechanical. So for those first 20 minutes or so, your nervous system is running a volume check, not a calorie count. A large salad and a large plate of fried food trigger a remarkably similar initial stretch signal if they take up similar space in your stomach. This is exactly why volume-based eating strategies work at all as a short-term appetite tool. They're borrowing this ancient mechanoreceptor system. But volume is only the opening move. Once digestion actually gets underway and nutrients start reaching the small intestine, a second chemical layer of signaling switches on, and this one is a lot more selective about what you actually ate. As partially digested food moves from the stomach into the small intestine, specialized cells lining the intestinal wall, called eye cells, start releasing a hormone called cholecystokinin, or CK, in direct response to fat and protein arriving in that stretch of gut. Unlike the stomach's volume sensors, CK is content-specific. It barely responds to a stretch of plain water moving through, but a rich, calorie-dense meal, especially one loaded with fat, sets off a much stronger release, which is part of why a fatty meal tends to feel more definitively done than a large glass of juice containing the same number of calories. The foundational research behind this hormone's role goes back to 1973, when a team of researchers demonstrated that injecting CK into rats caused them to stop eating well before they would have otherwise, even when food was still freely available. Again, this is an animal study, and appetite hormones don't always behave identically across species, but this finding opened up an entire field of research into gut-brain signaling that continues today. CK acts partly through the same vagal pathway that carries the stomach's stretch signals, meaning the two systems, mechanical and chemical, converge on the exact same neural relay station rather than competing separately for attention. Human research eventually caught up with the rat data. Researchers led by Debunking and colleagues in a study published in the journal Peptides in 2001 found that infusing CK directly into human volunteers suppressed both appetite ratings and actual food intake confirming that the same basic signaling pathway operates in people, even if the exact dose-response curve differs from what's seen in animals. So by roughly 30 to 40 minutes after that big meal, your body has already fired off 2 separate alarm systems attempting to slow you down. The bigger question, though, is what's physically happening to all that glucose and fat once it actually crosses into your bloodstream. And the very first organ in line to receive it might not be the one you'd expect. Here's a detail about your anatomy that most people never learn. Blood leaving your intestines doesn't go straight into general circulation. It travels first through a vessel called the hepatic portal vein, which routes it directly into the liver before it ever reaches your heart, your brain, or any other organ. This isn't a minor plumbing detail. It means the liver gets first access to every gram of glucose absorbed from that meal, well before your muscles, your brain, or your fat cells see any of it. Researchers confirmed exactly how this plays out using a clever, non-invasive technique called carbon-13 nuclear magnetic resonance spectroscopy, which can track glycogen concentration inside a living liver in real time. In a study published in the Journal of Clinical Investigation in 1996, Taylor and colleagues gave healthy volunteers a mixed meal and watched liver glycogen concentration climb steadily for roughly 5 hours afterward, representing a net synthesis of just over 28 grams of new glycogen, which worked out to close to 19% of the carbohydrate content of the meal itself. Insulin, released in response to rising blood glucose, is what flips on the enzyme responsible for this storage process, essentially giving the liver a green light to start packing glucose molecules together into long branching chains. But that green light only lasts so long because the liver's storage tank is small. Even fully topped up, a normal adult liver holds somewhere around 100 to 120 grams of glycogen, worth roughly 400 to 500 calories. For a meal carrying a genuine 1,000 calorie surplus, that tank fills up fast, sometimes within a couple of hours. And once it's full, the priority for incoming glucose shifts to a second storage site. One with a dramatically larger capacity, but a very different set of operating rules. Skeletal muscle, taken as a whole, dwarfs the liver in raw storage capacity, simply because there's so much more of it throughout your body. Using that same carbon-13 imaging technique on the calf muscle, Taylor and colleagues found, in a study published in the American Journal of Physiology in 1993, that between 26 and 35% of all the carbohydrate absorbed from a mixed meal ends up stored as glycogen inside muscle tissue, a substantially bigger share than what the liver manages to capture on its own. Insulin drives this process too, prompting glucose transporter proteins to move to the surface of muscle cells and pull glucose in from the bloodstream. Here's where the two storage sites part ways completely. Liver cells contain an enzyme called glucose-6-phosphatase, which lets them convert stored glycogen back into free glucose and release it into the bloodstream whenever blood sugar starts dropping. Muscle cells simply don't have this enzyme. Once glucose enters a muscle cell and gets converted into glycogen, it's essentially locked in, usable only by that muscle for its own local energy needs during contraction. It cannot be released back out to help feed your brain, your kidneys, or any other organ, no matter how badly they might need it later. This creates an interesting wrinkle for that 1,000 calorie meal, particularly if it was eaten while sitting at a desk or on a couch rather than after activity. Muscle glycogen stores tend to fill up faster and more completely when the muscle has recently been worked and partially depleted, since contraction itself pulls glucose transporters to the cell surface independent of insulin. A muscle that's already sitting close to full, because no exercise emptied it out beforehand, has less spare room to absorb the incoming surplus, which pushes even more of that glucose load toward whatever storage option comes next in line. This is probably the single most misunderstood step in the entire sequence. Most people picture excess carbohydrate getting converted into body fat almost immediately, as though the liver were some kind of assembly line running non-stop, turning sugar into fat the moment glycogen storage fills up. The actual biochemical pathway responsible for this conversion, called de novo lipogenesis, absolutely exists in humans. It's just far less active and far more conditional than most people assume. A landmark study by Atchison and colleagues, published in the American Journal of Clinical Nutrition in 1988, measured exactly what happens when healthy men were deliberately overfed massive amounts of carbohydrate under controlled conditions. Total glycogen storage capacity across the whole body worked out to roughly 15 grams per kilogram of body weight, meaning glycogen stores had to be pushed close to their absolute ceiling, requiring several consecutive days of substantial carbohydrate surplus before meaningful de novo lipogenesis kicked in at all. Only once that ceiling was reached did the researchers observe carbohydrate being converted into new fat in any significant quantity. That distinction matters enormously for a single meal. Unless someone had already been eating large carbohydrate surpluses for days beforehand, with glycogen stores already sitting near full. One isolated 1,000-calorie meal is unlikely to trigger much conversion into new fat through this particular pathway at all, which raises an obvious question. If it isn't mostly becoming glycogen, and it isn't mostly becoming new fat either, where does that surplus energy actually go in the hours that follow? That's exactly where this video heads next, starting with something your body does with digestion itself that quietly burns off more energy than most people realize. So far, you've seen that your stomach responds to volume before it responds to a single calorie, that your liver gets first pick of every gram of glucose, and that turning sugar into fat is a lot rarer than most people assume. Out of those three, which one changed how you think about a big meal the most? Drop it in the comments, because the next stretch covers something almost nobody talks about: the actual calorie cost of digesting the meal itself. Here's something most people never factor in when they picture extra calories. Digesting, absorbing, and storing food isn't free. It costs energy. This is called diet-induced thermogenesis, and it covers everything your body spends breaking a meal down into usable pieces, moving those pieces across intestinal walls, and packaging them for either immediate use or storage. In a review published in the journal Nutrition and Metabolism in 2004, Westerterp laid out just how consistent this cost is across a wide range of studies, generally landing somewhere between 5 and 15% of the energy content of a mixed meal. What's easy to miss is that this cost depends heavily on what the meal was actually made of. Protein sits at the top of the list, with digestion costing somewhere between 20 and 30% of the calories protein itself provides, largely because breaking down amino acids and rebuilding them into new proteins is metabolically expensive work. Carbohydrate comes in noticeably lower, in the range of 5 to 10%, and fat is the cheapest of all to process, often costing less than 3% of its own calorie content, since storing fat as fat requires very little additional conversion for a meal carrying a genuine 1,000-calorie surplus. This means anywhere from 50 to roughly 150 calories could get burned off purely through the mechanical and biochemical cost of processing it, depending heavily on how much protein was in the mix. That's a real, measurable dent, though clearly not enough on its own to erase the surplus. And digestion isn't the only unconscious process quietly working against that extra energy, because there's a second mechanism operating in the background that has almost nothing to do with what you ate, and everything to do with how your body behaves for the rest of the day. Beyond formal exercise, and beyond the basic cost of staying alive, there's a category of energy expenditure called non-exercise activity thermogenesis, or NEAT, which covers everything from fidgeting and shifting in a chair to maintaining posture, gesturing while talking, and simply moving around a kitchen. None of it feels like activity in any conscious sense. And yet, across a full day, it can add up to a substantial amount of burned energy, one that varies enormously from person to person. In a landmark study published in the journal Science in 1999, Levine and colleagues overfed 16 non-obese volunteers by 1,000 kilocalories a day for 8 weeks and tracked exactly where those extra calories went. 2/3 of the resulting rise in total energy expenditure came specifically from NEAT, rather than from formal exercise or from any conscious behavioral change. Even more strikingly, the degree to which each person's NEAT increased predicted, with a strong statistical correlation, roughly a tenfold difference in how much fat they ultimately stored, meaning some people almost completely offset the surplus through unconscious movement while others barely budged. For that 1,000 calorie meal from earlier, this means the outcome partly depends on something happening entirely below conscious awareness in the hours that follow. Some bodies will start fidgeting more, standing more often, pacing slightly more while on a phone call, all without any deliberate decision being made. Others simply won't shift into that mode at all. Either way, whatever energy survives digestion's toll and NEAT's toll eventually has to go somewhere. And for most of it, that destination is a very specific type of cell built for exactly this purpose. Whatever surplus energy makes it past all of the above eventually gets packaged into triglyceride molecules and stored in adipose tissue. But here's the detail that surprises most people: that storage happens almost entirely inside fat cells you already have, not new ones being manufactured on the spot. Researchers confirmed this using a genuinely clever technique involving carbon-14, a radioactive isotope that spiked in the atmosphere during nuclear bomb testing in the mid-20th century, and then got absorbed into the DNA of any cells forming at that time, functioning as a kind of biological timestamp. In a study published in the journal Nature in 2008, Spalding and colleagues used this carbon-14 signature to determine that the total number of fat cells in a person's body is essentially set during childhood and adolescence, and then remains remarkably constant throughout adulthood, even after significant weight loss or weight gain. What actually changes with weight fluctuation isn't the number of these cells, but their individual size, since existing adipocytes can each expand many times over as they fill with stored lipid. This means that one particular meal, even a large one, isn't creating new fat storage real estate in your body. It's making existing storage units swell slightly, a process that has real physical limits, since a fat cell can only stretch so far before it starts sending out distress signals of its own. For a single 1,000 calorie surplus, though, that ceiling is nowhere close to being tested. The bigger story, at least in the following few hours, is what's happening not inside those cells, but in the bloodstream carrying fat toward them in the first place. Dietary fat doesn't travel through the bloodstream on its own, since fat and water-based blood plasma don't mix. Instead, the intestine packages absorbed fat into particles called chylomicrons, which enter circulation through the lymphatic system before eventually reaching the bloodstream proper. If someone drew a blood sample a few hours after a substantial fat-containing meal, that sample would often look noticeably different from a fasting sample, sometimes visibly cloudy or milky, because of the sheer number of these fat-carrying particles suspended in it. Researchers led by Fielding, in a study published in the American Journal of Clinical Nutrition in 1996, tracked this process in detail and identified a distinct early peak in chylomicron triacylglycerol levels occurring roughly 50 to 60 minutes after a meal, followed by a broader elevation that can persist for several hours afterward, particularly if the meal carried a meaningful fat load. This entire process is referred to as postprandial lipemia, and it's a completely normal, expected physiological response, not a sign that anything has gone wrong. It's just not something most people ever think about when they picture where their food goes. For roughly 3 to 5 hours after that 1,000-calorie meal, a blood test wouldn't just show elevated blood sugar early on. It would also show a rising, then gradually falling, wave of circulating triglycerides working their way toward storage and toward tissues that can use them directly for fuel. And this wave doesn't necessarily finish its business by the time the day ends either. Your body runs on a strict prioritization system when it comes to which fuel gets burned first. In a study published in the American Journal of Clinical Nutrition in 1996, Jeb and colleagues used continuous whole-body calorimetry over 12 days to show that carbohydrate oxidation adjusts almost immediately to match carbohydrate intake, rising or falling within a day or two of a change in diet. Fat oxidation, by contrast, doesn't respond nearly as directly to how much fat is actually eaten. Instead, it gets pushed up or down largely as a leftover consequence of whatever carbohydrate oxidation is doing, meaning the body defends its carbohydrate balance first and treats fat balance as the flexible variable that absorbs the difference. This creates an interesting wrinkle for a single indulgent meal, one that doesn't fully resolve within the same day. In a study published in the British Journal of Nutrition in 2019, Dayton and colleagues overfed healthy young men by 50% above their energy needs for a single day, then tested them the following morning. Appetite the next day wasn't reduced at all compared to a normal eating day, and the triglyceride response to a standard test meal the following morning was measurably larger than it had been without the prior day's overfeeding. In other words, the metabolic ripple from one large meal doesn't necessarily settle by bedtime. Part of it appears to carry forward, quietly shaping how your body handles the very next meal you eat, even if you have no way of consciously sensing that carryover happening. Which brings up one final piece of the puzzle. Everything described up to this point assumes something that isn't actually true, that everyone processes an identical surplus meal in an identical way. Every mechanism covered in this video, the stomach's volume alarm, the liver and muscle storage priority, the rarity of new fat synthesis, digestion's calorie cost, unconscious fidgeting, and the way fat cells expand rather than multiply, plays out differently depending on the person sitting down to eat. Two people could consume the exact same 1,000-calorie surplus meal and end up in genuinely different metabolic places by the next morning. In a study published in the International Journal of Obesity in 2013, Schmidt and colleagues compared adults who were naturally resistant to weight gain against adults who were prone to it, feeding both groups the same 3-day overfeeding protocol. Total energy expenditure rose in both groups by a similar amount, but nighttime fuel oxidation patterns diverged sharply. The obesity-prone group showed a significant shift toward burning more carbohydrate and less fat overnight, a shift that essentially didn't happen in the obesity-resistant group at all. So when you picture that 1,000-calorie meal from the start of this video, there isn't a single universal answer for where it goes. For one person, more of it gets quietly burned off through fidgeting and movement they never consciously chose. For another, more of it lingers as circulating triglyceride or shifts into overnight fat storage instead. The biology involved, the stretch receptors, the hormones, the glycogen tanks, the digestion cost, the fat cells themselves, is the same for everyone. What differs is how each of those systems happens to be tuned in the particular body, sitting down to eat. This video covered a lot of ground, from the liver's storage priority to why turning sugar into fat is rarer than people think, to the fact that no two bodies handle the exact same surplus meal identically. Which one of those changed how you think about a big meal the most? Let us know in the comments below. If this kind of breakdown made you see something as ordinary as a meal a little differently, there's a lot more of this on the channel. Give it a like if you learned something new, subscribe if you want more of this, and send this to someone who's ever wondered what actually happens after they overate at dinner.